Hi, welcome to the 21st video in the IB Biology tutoring series provided by IBS. Today we're going to go over topic 4.1, Species, Communities, and Ecosystems. So without further ado, let's get into it. So for this topic, we're going to first go over a few definitions that are relevant to ecology, which is the big topic 4 for ecology. So ecology is the study of the relationships between organisms as well as their environment. And in this environment, and as well as these ecological systems, we're going to look at how energy is going to flow through these ecological systems, as well as the cycling of materials within the ecological system. So there are a few definitions or a few concepts that we're going to go over. The first of which is species. A species is a group of closely related organisms that are able to interbreed and produce fertile offspring. And when two different species do produce offspring by crossbreeding, these hybrids would then produce infertile offspring. And the reason why this occurs is something that we went over in the previous big topic, which is relevant to the number of genes and chromosomes. So, population. A population is a group of organisms of the same species that lives in the same area at the same time. And these organisms that live in different regions are unlikely to interbreed, and therefore we would interpret them as different populations. However, they are still classified as a same species if they can interbreed. So I realized that I missed a key definition that we have to go over, which is habitat. So let me just quickly write that out. So a habitat, a habitat is basically where the place an organism lives. So place where organisms lives. Okay, so there's that. I forgot to put it in the notes. So there's for habitat. For community, a community is a group of populations that lives and interacts with each other in an area. For ecosystem, an ecosystem are the interactions that exist between or within a community, as well as with its abiotic or non-living environment, such as different conditions such as light, pH, temperature, as well as soil. So for the biosphere is the interactions between all ecosystems. And the ecosphere is a closed ecosystem where it is self-sustainable and only light is needed to sustain this ecosystem. And finally, a mesocosm is a small-scale laboratory model to investigate the self-sustainability of ecosystems. And in turn, through using a mesocosm, we can control different variables and investigate how controlling or disturbing a factor can impact the ecosystem. So to help you sort of understand this, we would first have so actually these are all going in order of size, if you sort of think so. So population contains species, and then there's the community that contains those two, and then ecosystems further out. Biosphere is the biggest, stuff like that. So now that we've gone over some of the terms that are important in ecology, let's take a look at modes of nutrition, or how different species can intake nutrition. So we can subdivide them into two main groups. The first being autotrophs and heterotrophs. Autotrophs are organisms that are able to synthesize their own organic molecules from carbon dioxide, water, as well as inorganic nutrients, and therefore we would also refer to them as producers. You would know them to be basically plants, and the energy needed for this process is derived from sunlight or supplied from sunlight, and therefore they would be able to conduct photosynthesis. The second group would be heterotrophs, and heterotrophs are organisms that obtain their carbon compounds from other organisms, and they conduct this by either killing recently killed or consuming non-living remains. So under the umbrella of heterotrophs, we can further subdivide them into consumers, scavengers, and decomposers. So for consumers, consumers are able to intake their carbon compounds by ingesting organic molecules from living or recently killed organisms. For scavengers, they are able to ingest carbon compounds or organic molecules from feeding on dead and decaying carcasses instead of hunting living prey. For decomposers, they break down organic compounds into inorganic compounds, and therefore they mostly live in soil, and they can feed on the detritus, which are dead organisms as well as make waste material. So the stuff that they are consuming are completely dead. Under decomposers, we can further subdivide them into detritivores and saprotrophs. For detritivores, they ingest the organic molecules from non-living remnants of organisms, and they would conduct internal digestion. Whereas for saprotrophs, they would conduct external digestion, and they do this by releasing enzymes and absorbing the products that have been broken down by the enzymes. And for both detritivores and saprotrophs, by breaking down the organic compounds into inorganic compounds, they would then recycle the nutrients to the autotrophs, and autotrophs would then take these inorganic compounds and use them again, and etc. etc. And the final type of modes of nutrition would be mixotrophs. So they're under the same level as autotrophs and heterotrophs, but they are rarely talked about because there aren't a lot of them, and we will only see them in some unicellular organisms where they can use both forms of nutrition, both heterotrophic as well as autotrophic. However, this is dependent on the availability of the resources in their surrounding environment. So I sort of mentioned what we're going to talk about next, which is nutrient cycling. I sort of talked about how decomposers would break down they would recycle nutrients to the autotrophs. And basically what nutrient cycling is how the nutrients can be reused constantly from within the ecosystem itself. So the chemical elements are constantly recycled after they are used, and so therefore you would understand that the supply of these inorganic nutrients is finite, and we cannot spontaneously generate this inorganic nutrients from nothing. So therefore you would also understand that the inorganic nutrients are basically go in a cycle where they are changed from autotrophs to heterotrophs to decomposers as or saprotrophs, and then they go back into the autotrophs. So this cycle begins with autotrophs obtaining the inorganic nutrients from the air, water, as well as the soil, and then they convert it into an organic compound. Heterotrophs would then ingest this organic compound and use them for growth, respiration, and in turn release organic byproducts. When the organisms die, the saprotrophs would decompose the remains from the dead heterotroph, and they would free up inorganic materials into the soil. And the return of these inorganic materials into the soil can ensure that the autotrophs have a continuous supply of raw materials. And therefore, they would be able to build up more organic compounds, and the cycle begins anew. So let's talk about mesocosms. So mesocosms, I just gave a brief definition of it. So the main components of a mesocosm to simulate or mimic how a ecosystem or a ecosystem would look like. We would have different elements to that. The first of which being a light source, where we would have this would be where energy availability is determined. So we would be able to allow this light source would allow autotrophs within the mesocosm to produce carbon compounds as well as produce oxygen for other organisms to survive. We would also have to have saprotrophs to decompose the dead organic matter from within the mesocosms, and then we would recycle the nutrients, and therefore this would be a form of nutrient availability. And finally, we would also have consumers and detritivores to detoxify any harmful waste products, and therefore this would be where waste is recycled. So the three things that we need are energy availability, nutrient availability, and recycling the waste products. So we've neared the end of 4.1. Let's just quickly talk about ecological study, where it's basically how ecologic studies on distribution of species are conducted. So the presence of two species within a given environment will be dependent upon the potential interactions between these two species. And we can then perform a chi-square test for association between these two species. If the two species are typically found in the same habitat, we would see a positive association, whereas if the two species are typically not found in the same habitat, they would show a negative association. So this is an example of an ecological study, where we would look at whether two species are likely to appear together. So to conduct this, first we would have to sample we have to sample or count the number of the two species of interest, and we can do this by quadrant sampling, which is a form of random sampling. A quadrant is a square frame of a particular size. It could be arbitrary, but it would be set during conducting the study, and the species within the quadrants are identified and recorded, and we would then identify the abundance of a certain organism or a few species. However, there are a few limitations to quadrant sampling. The first being how we cannot use them to count fast-moving organisms, because then it would be really hard to determine whether they actually are within the quadrant itself. The organisms must be small because a large organism size would mean that it may be possible that it is hard to determine whether they're fully within the quadrant or not. And finally, we can only use it when the habitat is fairly uniform. Okay, so now that after conducting quadrant sampling or determining how many of a certain species is within the environment, we would then be able to conduct a chi-square test. And this chi-square test allows us to see if the distribution of the two species are associated or not. To do chi-square tests, we would use quadrant sampling to collect the data on whether a species is present or absent, and we would then compare the observed number to a mathematically expected number of species within the environment, and then we would compare the chi-square value to a critical value to show whether there's association between the two species. So chi-square test is conducted through 5 steps. The first is identifying the null hypothesis. The second is constructing a frequency table between the observed and the expected values of a specific species. The third step is to apply a chi-squared formula. Fourth, to determine the degree of freedom. And finally, 5, to identify the p-value. So I'm going to do these 5 steps here in this example. So first step, we would have to identify the null hypothesis. So that's done here. So for our null hypothesis, we would have no significant difference between the distribution of two species, and we would refer that to as a random distribution. Whereas for our alternative hypothesis, we would our alternative hypothesis is that there is a significant difference in distribution between two species, where the species are associated. So significant difference doesn't really mean that it's really different. It's just a sort of like a term that has been used for a long time. So that's step 1. We have identified the hypothesis for our chi-square test. So now let's go into the second step, where we would construct a table of frequencies. So I've already done this here. So initially you would only have the observed values, where you counted after conducting the quadrant sampling. So here are this is a hypothetical situation. So for species A, there are 10 present and 30 absent, and for species B, there are 10. So For species A, there are 10 absent and 10 present and 30 absent, whereas for species B, there are 10 present and 27 absent. Sorry, scratch what I just said. For species A and species B, crossing with species B, there are 10 squares that have both species A and species B, 30 squares that have only species B, 27 squares that have only species A, and 15 squares that don't have any species. So the squares would be the stuff that you would find that we would counted from the quadrat sampling. So we have the observed values here. Now let's calculate the expected value. So to calculate the expected frequency or expected number of species, we would first calculate the row total. So for example, I'm trying to calculate the expected value for squares that are present with both species. So we would take the row total, we would multiply it with the column total, and then we would divide it by the grand total. So the total number of squares or something like that. So the grand total here, I've already previously calculated this. It is 82. Okay, so 82. So then to calculate the expected values, it would be, for the first one, I'm just going to do one example. It would be 40 multiplied by 37 divided by 82, which would equal to 18.0. So that's the expected value. So for the sake of time, I've already done this before I recorded this video. So for this one, it's 22. For present species only species A, it is 19.0. And for all species missing, it will be 23.0. Okay, so we have the expected values. So after we've constructed our table of frequencies, we will apply the chi-squared formula. So the chi-squared formula is here, which is basically the sum of it is observed minus expected squared, observed minus expected, and we will square that value, dividing by expected frequency, and then we would add up for all of them. We would add up for each cross here. So again, let's use the first example. So our observed here for squares that are present for both species B and species A, it is 10 minus 18 squared divided by 18, which would equal to 64 divided by 18, which equals to 3.56. Okay, so again, for the sake of time, I've done the remaining ones. So next is 2.91, this is 3.37, and this is 2.78. Okay, so we have all of the chi-squared values for each cross. So we would then add all of these values together. So for the chi-squared value, it is 3.56 plus 2.91 plus 3.37 plus 2.78, which would equal to 12.6. So this is the chi-squared value. Now that we've calculated that, we're going to have to determine the degree of freedom. So we're not done yet just by calculating the chi-square value. We need to use this chi-square value to determine whether it is significantly different or not, if there's a significant difference or not. So the fourth step, determining the degree of freedom. To determine that, we would the degree of freedom is m minus 1 multiplied by n minus 1. So m is the number of rows and n is the number of columns. So in our case, it would be 2 we have 2 columns, and we have 2 rows. Sorry, 2 rows and 2 columns, which is still just 1. So df is always 1 when we're only comparing 2 species. So the final step is to identify the p-value. So I've drawn here what is basically a statistics table, and usually this thing is given in the exam if they do test you on chi-square stuff. But for the sake of our notes here, I've drawn the whole thing out. I've drawn a portion of the relevant portion. So we would take our value here, 12.6, and we would look at whether it fit, where it would fall under in this, in this row. So we can see that 12.6 is beyond 6.635, and what this means is that our probability, our p-value is beyond 0.01. So this is p-values. So in our case, 12.6, the p-value is smaller than 0.01. And for anything to be, for a value to be considered statistically significant, we would only have to, the p-value has to be, for statistical significance, p-value has to be less than 0.05. So any value that lies along here would be lies along beyond would mean that it is statistically significant. It has to be smaller than the value has to be smaller than 3.841 in this case. So our calculated value is 12.6, so it is much bigger. So statistical significance, p-value has to be smaller than 0.05, and the calculated value has to be larger than 3.841. And ours is 12.6, making it statistically significant. By being statistically significant, we can reject the null hypothesis. So what if the question asks you to conduct the chi-square test, what you would write down is that we have a statistically significant. We would reject the null hypothesis, and that there is a significant difference in the distribution between species A and species B. And so if it were in the actual exam, you wouldn't just write species A and species B. You would actually fit in whatever species or thing that they're trying to test on. So that's how you would conduct a chi-square test. I really spent a lot of time on this because I think that is really important. And by finishing that also concludes topic 4.1. I'm not going to give an essay question on a chi-square test because I think that's really in depth on what I want to teach. But there are some other essay questions. So the first being, describe how dendritivores obtain nutrition and the effects they have in ecosystems. Okay, so that's for 4 points. So there are quite a few points that you can talk about for that. It's important to remember that you have to both talk about how they obtain nutrition as well as how what they do can affect the ecosystem. So this question will sort of play into nutrient cycling as well as modes of nutrition specifically for nutritivores. Okay, so the second question that I would talk about, I would like you to finish, is compare the way in which autotrophic, heterotrophic, and saprotrophic organisms obtain energy for 6 points. Now again, compare and contrast. Comparing would mean that if there are any similarities, you should also mention that. But however, it is also good that you sort of find some of the common areas that these three modes of nutrition would share. So for example, where they obtain the carbon compounds, how they would do that, etc., etc. So 6 points, you can divide them into 2 points for each type of mode of nutrition, and that would usually give you and 2 points for each. That's you use 6 points for 3 of them. So that's the end of topic 4.1. We're going to see you in topic 4.2.